Hello, everyone, and welcome to our panel session this afternoon. It's entitled Personalizing Cardiovascular Therapies to Address the Underlying Causes of Disease. My name is Monica Shah, and I am the head of the IQVIA Cell and Gene Therapy Center of Excellence. I'm also a heart failure cardiologist, so it comes as no surprise that I think that this area is one of the most exciting, if still uncharted areas in all of cell and gene therapy. So as everyone knows, cardiovascular disease is a global epidemic and represents some of the most lethal and costliest diseases in the world. And while there have been great strides in cardiovascular medicine, and arguably the field is perhaps the most sophisticated in terms of clinical trials, cardiovascular cell and gene therapy is still a very small fraction of the entire cell and gene therapy landscape, as you can see here in the upper right-hand corner. I think I would say that, many would say actually, that cardiovascular cell and gene therapy is still in its early days, and our panel here, you can share your thoughts on this, but I think we're still waiting for our first home run in the field. Today I'm really delighted to welcome a very distinguished panel who I consider to really be pioneers in the field of cell and gene therapy cardiovascular disease, blazing the way in often unmapped territory. So today we're going to be talking about their unique perspectives on some of the opportunities as well as the challenges in this field and some of the important lessons that they've learned in their journey in clinical development in cardiovascular cell and gene therapy. So with that, I'm going to ask each of our panelists to introduce themselves, maybe talk a little bit about your product and your science. And then actually most importantly, I want to ask each one of you to share why you're so passionate about cardiovascular cell and gene therapy. I mean, there's a lot, perhaps more, maybe easier spaces in cell and gene to carve out, and so why have you selected this area? So with that, let me turn it to you, Faraz.
Hi, thanks, Monica, and thanks to the organizers for putting together this great panel. I'm Faraz Ali. I'm the CEO of Tenaya Therapeutics. Tenaya Therapeutics is about a 100-person preclinical stage company. We recently went public. We're 100% focused on heart disease. We're advancing 3 platforms, gene therapy, cellular regeneration, and precision medicine. A lot of what we do involves viral vectors and gene therapy, but we're officially modality agnostic, and in fact, we also have small molecules in our portfolio. We're focusing on both genetically defined rare forms of heart disease as well as more prevalent forms. And yes, this is not for the faint of heart, and we're incredibly excited by the field. We think that the time is now for novel approaches for heart disease, and it's really what brings me into work every morning is just the opportunity to do good. There's tremendous unmet need beyond just the fact that it's the leading cause of death in the world, more than all oncology combined, but we are within that. There are genetic forms of disease where just truly just incredible unmet need, including children who are dying within the first few weeks and months of life, where the science has arrived to do something that could save their lives, and that's a good enough reason alone to do what we're doing.
Great. Joachim.
Yeah, no, I'm Joachim Frobis. I'm the Chief Development Officer for Blue Rock Therapeutics. Blue Rock, as most of you know, is a company fully owned by Bayer, but very much independent, developing cell therapies, cell plus gene therapies in multiple disease areas. Our lead area is in neurology, where we are in the clinic, cardiac and immunology, ophthalmology close behind. And I'm very excited today. I started out my career in the cardiovascular field and worked on more traditional approaches in that field, small molecules, biologics. And to me, it was just illogical to join Blue Rock a couple of years ago, because when you do classical therapy, you realize that, especially in heart failure, late-stage heart failure, where we are active in, you're dealing with ischemic tissue, and there's very limited opportunity for classical pharmacological agents, and so being able to do regenerative medicine in taking the cell therapy approach is just a culmination and a great opportunity to bring something to patients that they so desperately need.
Great, thank you. Jordan.
Great. So my name is Jordan Lancaster. I'm the Chief Executive Officer for Avery Therapeutics. Avery is developing tissue-engineered approaches for cardiovascular disease. We've developed a delivery platform that we call MyCardia. Our journey really started in the development of MyCardi was really around heart failure of ischemic etiologies, and we've subsequently explored and now have pilot programs for looking at that platform for treating varying arrhythmias. I think that my passions around cardiovascular, I've always just really been interested in the heart, and cardiovascular regenerative therapies have really just remained elusive for the entire field. And I really think that the being able to contribute to this field, really be able to shift that and provide transformative care to patients, if I can have any role in that, that's a tremendous opportunity. So just really excited for what we can bring to the table, the opportunities that we have for potentially treating patients.
BJ.
Okay, I'm BJ Lehman. I'm from Athersys. We've been active in the cardiovascular field for about 15 years now. I define it very broadly. We will talk about AMI, which has been our lead program. We've done clinical research in that area, but we've done work in other areas as well. PVD, we've looked at chronic ischemia. We've done some preclinical work in heart failure models. We've looked at wound healing in an adjacent area. We've been active in ischemic stroke as well. We're working with a cell product, a stromal cell product that's, we believe, driving value through the paracrine effects. Modulates the inflammation associated with cardiovascular disease and certainly contributes to the generation of new vasculature. We're very excited to be involved in the area. We think we have something pretty special to offer the cardiovascular field. I think we can make a difference and change the way medicine works. But one of the real reasons I'm excited about this is we have the chance to really help people. I have heart disease in my own family, so I understand this. And I can see what this does to quality of life. And I think with the therapies we're talking about today, we have a chance to change that.
Thank you. Michael.
Yeah, good afternoon. Thanks for coming and thanks for the opportunity. Mike Scott, I'm at Novo Nordisk in the cell therapy unit. We're an arm's length division of Novo Nordisk, and we're focused on heart failure, Parkinson's disease, dry AMD, and type 1 diabetes. No surprise on the last one there. I've been involved in medical device development and cell therapy development to treat cardiac disease since 1992. And since then, at the time, almost 30 years ago, the mortality at 5 years after diagnosis was around 50%. And if you look at that number today, it's about the same, despite all of the advances that we have made as an industry. So my passion is we have to move the needle on this disease. And I have a personal connection, starting with losing my grandfather at age 12 and 2 uncles in the last 24 months. So my mom went in a 6.5-hour on-pump operation over Christmas last year. So I am very passionate about this.
Well, that's great. Thank you for sharing those personal connections. It really, it's why we do all of this, so thank you. So our first question to the panel is going to actually focus on mechanisms. And, there's a few theories of how cell therapies work in cardiovascular disease. Differentiation of the cells to cardiac cells or myocytes, formation of new blood vessels or angiogenesis, and then, of course, the paracrine effect. So maybe I could ask BJ to start this question or this answer. So what do you think are the key mechanisms of cell therapies in general? And specifically for your therapy, what's sort of unique about your science?
I touched on it with respect to our cell products, and I'll come back to that in a second. I think each of the mechanisms you talk about can be very relevant in treating heart disease. I think it depends on the indication. Some of the mechanisms are going to be more relevant for certain indications. So for us, we've selected AMI as a target indication precisely because we have an opportunity to intervene early in the disease progression and affect the trajectory following the MI. And with a mechanism that we believe is at work here, we have the opportunity not only to dial down the inflammatory response, which leads to scar formation, et cetera, we also have the ability to support revascularization. And we believe that's pretty critical to the progress post-MI. We think it has lasting effects. So based on the mechanism we have with the MAPC product, we've selected MI as an early entrant for us.
Great. Joachim, do you want to answer that question?
Sure. No. So we are at Blue Rock, we are focusing on advanced stage 3, 4 heart failure, post-MI, massive MIs with large ischemic areas in the heart. And our approach has been to really rejuvenate and regenerate that tissue by seeding and bringing new cardiomyocytes into that area. Now, we say cardiomyocytes, but there are different flavors and different types of cardiomyocytes, and we spend together with our collaborators a lot of effort to really, at the very molecular genetic level, to understand the type of cardiomyocyte that it takes that needs to go into that part of the heart, and to translate that into a bioprocess to make exactly those cells, overcome many of the challenges from manufacturing very large numbers of cells, dealing with engrafting of the cells, physiological, electrophysiological engraftment. So at the bottom line, we're really trying to put back the authentic cardiomyocyte into that tissue to become basically one with the existing heart and bit in sync.
Jordan.
Yeah, so I think that mechanism is a great question. I think and my perspective, really looking at heart failure of ischemic etiology, our product is really quite interesting. While it's an engineered product, we are going to be working more within that secretome spectrum of the treatment, where we're really not anticipating our cells to have any real longevity. So an interesting differentiator between our technologies. But we know that our product, really just the unique nature of it, gives off dozens of different components within a component that we really just broadly refer to as the secretome. And the product really is unique in that we've had this developed syncytium of tissue that can be very easily implanted and deployed, and interacts very intimately with the underlying host tissue, and has a very nice dwell time before it's ultimately reabsorbed. And so it's a really interesting and just sort of a different approach, specifically, and that's heart failure of ischemic etiology, and we've got some tremendous outcomes in regards to preclinical data and large animal models that are really compelling in regards to being able to move the needle. But we are more of a secretome-based approach for heart failure.
And last but not least, Michael.
So our approach is similar to Blue Rock's, focused on heart failure patients. And by the time you're diagnosed with heart failure, you're typically well beyond the acute phase of disease. So it may be that paracrine effects may not be as beneficial. So our target is remuscularization with, of course, potential paracrine benefits in this advanced population with various relevant etiologies. Most people don't necessarily understand that heart failure is not a disease. It's a family of diseases with a lot of underlying etiologies, and the one hammer won't work for all of those nails. So we do have to be, I think, quite cautious and thoughtful about how we apply these different technologies, and there's plenty of opportunity and space in this business for these various different approaches.
I think that's a really great point. We've often approached heart failure as sort of this grab bag of, it's one disease and throw multiple different therapies at it. And I think that's why we've had kind of mixed results in the field. So I think you've raised a really good point, Mike. Okay, so let's move to the next one. And Faraz, this one is for you. We said in the beginning that we're still waiting for our home run in cardiovascular cell and gene therapy. And so there's a lot of proposed reasons for the somewhat mixed results of some of our trials. They say cell type, cell dose, wrong targets, missed targets, the route and frequency of cell and gene therapy, administration, and the durability of our therapy. So maybe you could comment on what you see as the key challenges in developing effective therapies in this space.
Yeah, thanks. Excellent question. First, I'll just say we are a bit of a dark horse on this panel in that we're not advancing a cell therapy approach. We do have a cellular regeneration approach, which is completely distinct. It's actually in vivo regeneration of cardiomyocytes using viral vector approaches to reprogram fibroblastic cardiomyocytes. So that's a totally different approach. The mechanism will be, to be determined, but very different. But in the field of gene therapy, so not cellular regeneration, but gene therapy, yes, there's this checkered history, and I think there's a few. And the most recent example that perhaps most people remember is Celadon, which advanced a product to use AAV gene therapy to add the SERCA gene to the myocardium and try to address heart failure. And that had early promising results, and then they flamed out, I think, in phase 2. But we learned a lot through that experience, and I think the field has advanced a lot since then. So I think a couple of things that have changed since then that make the field, puts it on firmer foundation than back when that last attempt was made. One is viral vectors. First generation efforts were using AAV1, AV2, Celadon used AAV1, but now we know that other vectors like AV8 or 9 have significantly better tropism for the heart and do a significantly better job penetrating the myocardium. So, vectors certainly help manufacturing. I think, back in the day, we didn't appreciate how important it was to really lock down your process and how changes in process can really fundamentally change the product. That's what happened with, this is all in the public domain, but Celadon changed their process from phase 1 to phase 2, and that resulted in a different empty to full capsid ratio and other differences in the product attributes. We now know that that's a cardinal sin. You don't do that. You want to really lock down your process and not make too many changes along the way. So manufacturing technologies have also changed. We now have cardiac-specific promoters, and we have the ability to improve on cardiac-specific promoters to really exquisitely ensure expression within the heart versus other organs. And even within the heart, we now have the ability to target, say, the cardiac fibroblasts versus the cardiomyocytes, and to dial up or down the expression of a gene of interest within the heart. So all of those things were just simply not possible back when people were doing studies a decade or so ago. But the most important thing, in addition to those scientific advances, I think, is the genetics of heart disease. Kind of building on the point that you were making, we can now peel back the onion and take heart failure, which is the leading cause of death in the world, but really start isolating genetics. We have a lot more genetic insight than we did a decade ago. And with that, we have the opportunity to then develop therapies that are more precisely targeted to specific genes. So, back in the day, people would talk about hypertrophic cardiomyopathy, dilated cardiomyopathy, arrhythmogenic cardiomyopathy. But now we can actually talk about MYBPC3 mutations that are the leading genetic cause of hypertrophic cardiomyopathy, or PKP2 mutations that are the leading genetic cause of arrhythmogenic cardiomyopathy. And then that opens up the door to understanding whether a gene transfer or gene editing approach could work. And so that's just something, that's an insight that just didn't exist about a decade ago when earlier efforts were made. So I think a lot of early gene therapy and cell therapy efforts have focused mostly on prevalent indications. And now with companies like Rocket Pharmaceuticals and now Tenaya and others, we're beginning to go after more rare, but still prevalent, but more rare, genetically defined forms of heart disease. And that makes all the difference. You can imagine getting early proof of concept data in a small number of patients. And with a lock and key gene therapy approach, there's a much higher probability of technical success than previously existed.
Thanks very much. That's great. Joachim, what are your thoughts on what you think might be, sort of the key challenges in developing effective therapies?
Maybe I just want to expand a little bit on, for us, was talking about, because in a way we are approaching the issue from two different angles, and both obviously well thought through. And as he pointed out, the big difference to me is really, it is that monogenic angle versus the whole cell approach. And we know, we've seen a lot of these approaches, be it angiogenic or SERCA channel approach, targets that clearly are valuable, but some of the failures have taken place exactly there, and mostly because probably the match wasn't there. And I know that Tenaya is taking a different approach, really making the match between the patient, identifying them for the genetic defect and trying to correct that. And so we're going more broad into that larger ischemic area, and which allows us to be a little bit more agnostic, if you want, when it comes to the patient, and that's really what's so different here. The challenges along with that, of course, now you're putting cells into an ischemic area, and you have to allow these, you have to work to make these cells engraft, get all the physiological supply, angiogenesis, all these good things need to still take place. You want the cells to not just engraft, but engraft in an electrophysiological way so that you have a healthy heart rhythm. and you have to, a person with a severe post-myocardial heart has lost on the order of 1 billion cells. That's a lot of cells that you need to put back. So there are significant challenges, absolutely, to put those back into the beating heart, which is probably the last challenge, right? To delivery of those cells into the beating heart.
Thank you. Yeah. BJ, your thoughts on this?
Yeah, no, sure. I'm going to talk about the delivery in a second. I think Joachim's onto something that's very important. But a couple of things. First, there are common challenges for all cell therapies that are relevant here. You've got to be able to demonstrate that your cell has the targeted effect, and you've got to be able to characterize that well. Very critical from a regulatory perspective. You've got to be able to manufacture the product. It's not insignificant for us and for anybody in the area. I think delivery is critical, though. We spent a lot of time in our early days trying to define what the right delivery mechanism was. We've considered IV delivery. Remember, we have a product that's delivering an effect through the proteins and other factors that are being generated. So you could imagine an IV product working in this area, but you have to deliver enough therapy in the area to have the impact you want. We've looked at direct delivery, so direct into the heart, maybe tied together with some sort of surgery or something like that. That seems to be effective in the models, but it's not very practical if you want to treat a lot of patients, particularly in AMI. So we focused on the delivery mechanisms available at the time. This is in the late 2000s, roughly, so there's been a lot of technology development since then. And we evaluated a variety of different mechanisms, and we ended up selecting one that uses a micro syringe that delivers the product into the layer, adventitial layer around the heart. And for us, that was very helpful because it created a residence time for our product. And we were able to demonstrate in large animal models that the cells would stay embedded for quite some number of days, which, if you look at it from an IV perspective, just does not happen. You lose the cells from the heart in a single passage, essentially. So that was a very important early part of our development. I think it's a key challenge, and I think it's critical to success. And with the approach we've taken, we've developed an approach where you can deliver a relatively, I call it modest number of cells, 50 to 100 million cells, and we believe have impact. We've had some pretty promising signals from the early clinical development, and I think it's got potential, but that's critical. Another area that is a big challenge, and one we've struggled with in clinical development, has to do with the target endpoints. So if we think ahead and we think about what's going to be approvable, we kind of bring that back into the early clinical world, trying to understand the surrogates or the measures that are going to give us an indicator about outcomes, right? That's the gold standard in the area. Our expectation has always been that to get approval, that's going to be part of the package. We hope that there are other surrogate endpoints that develop over time. I think that's possible. But we spent a lot of time thinking about that. And so in one of our studies, we invested fairly heavily in imaging to understand the impact on perfusion. But that's very tricky to do and pull off in a study, particularly when you've got to evaluate that over time. And you have to have these patients come back in and be available and be imaged. So it can be very tricky. This development can be very tricky if you want to demonstrate in a clinical setting that benefit that ultimately is going to translate into clinical outcomes.
I think you've raised two really good points on why it's so difficult to do trials in this space. First, you've got to find the right investigators who have that technical skill to be able to deliver the therapy. And then if you're doing all these imaging and all these additional studies, to be able to find those types of institutions that are capable and have the infrastructure to do that. So I think these are part of the challenge that's unique to cardiovascular cell and gene therapy clinical development. Jordan, you deliver your therapy through a surgical intervention. And so maybe you could share a little bit about that and what are some of the unique challenges that you're seeing there?
Yeah, absolutely. Our development story is actually quite interesting. It started, kind of on the time frame that BJ was talking about, seeing early cell therapies kind of advancing to clinic. And we were in a very clinician-heavy group at the time, and we were having constant discussions and debates about the right types of cell, when should you be delivering those and how you should be delivering those. And we kind of, started breaking that apart and really did a lot of work just playing foundationally on bringing different types of cells together, implanting them and looking for physiologic outcomes, and tinkering and playing with that and leveraging certain things that we discovered along the way, like how to make a very angiogenic graft, and then leveraging that to ultimately get to a point where we could combine cells and have very profound structural changes of the heart. We're really looking at specific things like volumes and masses, and more sophisticated things like in-systolic elastance or in-diastolic pressure volume relationships. And I think that, from our perspective, there were a lot of challenges around, less so on the development of the cell, particularly for us, but really the biomaterial component of how you're going to manufacture, what is the right biomaterial components that allow you to do that manufacturing in vitro. We are also unique in the ability to cryopreserve our product, so being able to do that and then distribute and having a product that works really well in the clinic. We have an example when we were doing one of our first swine procedures. We had upscaled the technology, handed it to the CT surgeon. He goes to implant it, and he was pissed. The product did not handle well. And so we tinkered with this and ultimately developed a substrate that doesn't fold on itself, doesn't stick, but has really good drapability. I talked him into coming back, showed up. Next time he brought a friend. So we knew we were onto something of really solving that challenge of how you're delivering, and that biomaterial really played a role. So it's kind of a fun development story, and for us the challenge was really the biomaterial. From a surgical application perspective, we can do a de novo approach. Again, I'm referring to heart failure. A de novo approach or adjunct to coronary artery bypass grafting, CABG, or LVAD. From a de novo approach, we have about 6 different surgical approaches we can use, 4 of which are classified as minimally invasive, and the technology is really well suited for video-assisted surgeries, robotics in particular, so small 1.5-inch working ports, so low burden on the patient, very quick implants of about 30 minutes in our preclinical studies, so very, very straightforward.
Thank you. Mike, I'm going to ask you, you are a device specialist and as well as a cell therapy specialist, and you clearly have had to navigate some of those unusual pathways, or maybe multifactorial pathways, if you will, with approvals on both in multiple parts of many agencies. So maybe you could talk a little bit about some of the challenges from that perspective.
Well, the first level is, what is the classification of your product, and is it a combination product officially, informally, or not? Irrespective, you have to meet all of the regulations. So if it's a device, you've got to meet CDRH's requirements. If it's a biologic, then it's CBER. You start combining those, and then you have to deal with all of the interfaces and interactions between those. For example, your delivery device can have a direct impact on the health of your delivered cells, because you're going to induce shear stresses, for example. You're going to have impact on physical and chemical environment. And at the same time, your delivery device could get clogged up by cells settling out. So you now have these interactions between different medical device products that you've got to deal with. And the regulators will not allow a device company to go to, say, FDA or EMA or anywhere else in the world and get a label allowance for delivery of a cell or gene therapy. They'll say, what cell therapy, what gene therapy, because we have seen in the past have been burned by programs where these interactions were not properly characterized. So this is a challenge for our industry, because in many cases you have a cell therapy-based company that approaches this problem and says, we've got the biological magic, this active biological ingredient that is going to solve the problem. And there isn't a full recognition that that is not your product, that's your ABI, if I can steal that term. And you have to think of this as a multifactorial combination product. So you think, okay, for a product like ours where we want to deliver transendocardially, we'll need a delivery catheter and a steerable sheath and a dose control device. May start as a syringe, something that simple. Then we're going to need our cellular drug product and the formulation to inject. Are we done? I'm at 5. No, we need to know where that catheter is in the beating heart real time during the procedure. So now we need to take real-time 4D imaging technology, that's 3D real-time, and combine that with our surgical plan so that we know where to deliver the dose, so that we can see in real time. So is your head spinning yet? Because now I think I'm up to 8 different technologies that all have to work at the same time in the same operative suite to successfully deliver in a safe and efficacious manner.
Thank you. Okay, so let's move on to a little bit more about clinical development. As we all know, cardiovascular disease is polygenic, often polygenic, multifactorial, and so therapies will need to address multiple failing mechanisms in order to be effective. However, there's a lot to learn from our experience in cell and gene therapy from other fields, other more established fields. And so maybe I can ask Faraz to address this question first. How's the paradigm of cardiovascular cell and gene therapy different, and how is it the same? And what are the biggest lessons that we can learn from, let's say, oncology or rare diseases for cardiovascular disease?
I think you've given part of the answer, right? Because I think the opportunity here, kind of continuing where I left off with the better insight in terms of genetics of heart disease. So yes, there are more complex multifactorial forms of heart disease, but we at Tenaya are initially focusing on where the genetics are clear. And what that means is that we can start applying precision medicine approaches that have worked very well in oncology and start applying them to cardiology. Now, we're not alone in that. I mean, I think there's been some movement. Some of you may be familiar with, Pfizer had a program for ATTR cardiomyopathy. They had an approval back in 2019. That was a relatively small phase 3. It was 200 or between 200 and 300 patients based on a composite endpoint. And so there was some novelty there. And they got an approval for a rare genetic cardiomyopathy. Now everybody is waiting with bated breath, but there's hope that MyoKardia's product, mavacamten, now part of BMS for hypertrophic cardiomyopathy, another precision medicine approach, not maybe based on genetics, but based on mechanism, in hypertrophic cardiomyopathy, that also with a 200, 300-person study, again with a multiple endpoints there, some functional, some quality of life, in addition to looking at hemodynamic and stress markers and echo parameters, that could have an approval as early as early next year. So I think we're in an era where there's some willingness by the regulators and by physicians to consider novel approaches to heart disease drug development. And now in the case of rare genetic disorders where it's a lock and key approach, which is some of the early gene therapy approaches that we're advancing, again, we'll have to do some of the same things. We'll have to look at stress markers of stress, echo markers. We'll have to look at quality of life. But we don't anticipate that we're going to have to demonstrate survival over a long term and a long period in order to get an approval, because in many cases these are very severe, small populations, very severe, they're dying early, and the genetics are so clear and the therapeutic intervention is so matched to the genetics that we think that the regulators will be open to approval based on other endpoints, consistent with what has been true for other gene therapy products. So I think that opens up the paradigm to doing things differently, doing things more efficiently, allows companies to move quicker, get proof of concept earlier, and be able to take these programs further along in development on our own. So I won't speak to the polygenic ones, that's sort of the next horizon, but I think there's a great unmet need and great opportunity with monogenic disorders. Now, some of them will be gene transfer, some of them will be gene editing or gene silencing, but the closer we can get to understanding the mechanism of action and then matching our therapy to that precise mechanism of the disease, then we can aspire for smaller studies with bigger effect size, which is very different where historically cardiovascular drug development has been, where you're running outcome studies for 5,000 or 8,000 patients, generally with small molecules, and looking for a pretty small effect size and looking at survival endpoints. Very expensive, very high risk, very high reward, overall low probability of technical success. So I think we're entering a new era where some new things are possible, at least in the field of gene therapy of the kind that Tenaya is focused on today.
Great, thank you. So BJ, you are focused on acute MI, heart failure. What are some of the parallels that you potentially see with, let's say, oncology or some of the other diseases that we have seen more of a history of cell therapy in?
Yeah, I think in AMI, actually, the opportunities or comparisons are much more limited. I think for us, is in the approaches they're taking, they're very smart, and I think it offers opportunities for very efficient development and ultimately early approvals. In AMI, I think the expectation is going to be different, frankly. And we've actually spent a lot of time trying to narrow the population we're going after. We started with STEMI, as big as you can get. Tricky area, standard of care is rapidly changing. We could have impact, but it'd be pretty tricky to get this through development. You focus on end-STEMI, closer to heart failure, really focusing on microvascular disease. So you're starting to bring that together, the therapy with the real disease here. But we're never going to get to that very targeted patient population with the approach we're taking. So our expectation is much of the work that's been done more generally in cardiovascular disease is probably more relevant to us than what's been done more recently in cell therapies, generally speaking, and gene therapy as well. I wish we had those opportunities, but I think we've got to work with what we have.
Well, what do you think about the idea of actually narrowing the population so that, there's two schools of thought here. You go broad and you use your cell therapy in everyone who has an acute MI, or you really look for those, targeted populations, or you look for end-stage heart failure patients that are on their way to LVAD. So you're a very circumscribed population. Maybe, Jordan, you can address that question first, and then we can go down to Mike.
Yeah, excuse me. I think it's a really complex component. I think that, the challenges of trying to find, the correlation to oncology, really there's, what biomarkers are you going to be looking at? The disease is really heterogeneous. You're going to have comorbidities. It's just a very challenging component to address. And I think, the way we kind of look at it, just to cast a very broad net here, is, our preclinical models. I think that we can all agree that there's fairly decent preclinical models of heart failure. They're going to be specific, and they're going to have, their own component, and really understanding the model, how long you're letting the disease develop, how you're treating, when your technology starts interfacing, you're starting to see that signal, capturing a lot of information there, trying to learn as much information from these previous clinical trials as possible, really learn from their mistakes or shortcomings, what actually, was easy data to capture, and really just talk to your clinician base to really, think logically through your study. But I think it's really hard to look for any type of, especially, and I'm really coming from the heart failure perspective, biomarkers, those types of components are just really challenging. So, I agree, it'd be great to drive that population down, be selective about it, but you kind of have to, what data do you have, what's been happened before, and, talk to the experts about what they think was, impactful for that next stage.
Great, thank you. Mike?
Well, I'm not sure that I'm qualified to talk about direct correlations with the cancer area, but certainly there have been changes and some fairly clear direction in the cardiovascular products development industry, whether it be drug or medical devices, over the last 2 decades, where functional endpoints are just not accepted in clinical trials. They can be part of complex composite endpoints, but generally they're looking for clinical endpoints, meaningful improvements in quality of life, as an example. Because there have been way too many products that have gotten through early approval or early clinical trials and even approved that may improve short-term metrics around function but have a negative effect on longevity, for example, or quality of life. So the bar is higher. We're challenged with trying to meet clinical outcome goals that a lot of the products that are currently used as practice of medicine have never had to meet. So it's a tough bar when you're bringing a brand new technology in, and the bar is higher than it ever has been historically.
Great, well, thank you. So, we talked a little bit about manufacturing earlier, and of course that's been a big theme of this meeting. But one of the things that, I think is, again, unique about cardiovascular disease is if you're going to be successful, you're going to have to manufacture really at a large scale. So again, I'm going to maybe ask BJ and then maybe Joachim to address this question. So how do you address the scalability question in cardiovascular cell and gene therapy?
Yeah, it's a critical question. I mean, we're also developing in ischemic stroke, massive product need. Same is true in cardiovascular disease. You can do the math, 100 million cells times lots of patients, it's a lot of product. And so we've spent quite a lot of time trying to develop a product ultimately designed for commercial purposes. And that's carried us from, the standard two-dimensional cell factory-based culture approaches, which are very inefficient, as we all know, to bioreactors and scalable bioreactors. And there are all sorts of challenges around that, as we all know. I think folks have mentioned a couple of things. I mean, not only do you have to make a lot of product, you've got to be able to process a lot of product, you've got to be able to freeze down a lot of product, you've got to make it easy to be used at the clinical site, and you've got to manage the cold chain between the manufacturing plant and the sites. And we've spent a lot of time on that. I think we've developed a large-scale manufacturing platform that's suitable for some of the indications we're going after, but it was not without substantial investment. And it's been challenging, but I think we are getting there, and I think we could serve a market like AMI if we are able to develop a product successfully.
Thank you. Joachim?
Yeah, no, I think the manufacturing, just like device development, the process development, understanding your product, and so on, there's so many things that you have to work on in parallel, because the moment you go into a clinic, the moment you go into late stage or into commercialization, you need it all. You can't do it, and so you have to do substantial investment. And Blue Rock, one of the attractions about the company was really to recognize and the willingness to do this. So very early on, we set up our GMP facility in Toronto that actually supplies clinical material for even ongoing trials already in the Parkinson field. And the acquisition by Bayer, of course, allowed us to go even bigger. We are building now a commercial size plant. So that's not just a suite anymore, it's a plant in Berkeley, California. That's happening right now. Again, you have to do these things, everything takes time. And it's very exciting to see, but it can be done.
Wonderful. Okay, so I think another challenge that we have in cardiovascular disease, I don't mean to just focus on the challenges because there's a lot of good here, but the other challenge that we have is there's a lot of really cost-effective therapies that are out there. Granted, they don't, hit the home run, but they're, beta blockers, ACE inhibitors, Entresto, even, some of the new Vericiguat. So we have a lot of new things that are out there. How do we make the calculation? What kinds of things do we need to show to payers, to patients, to physicians, to show that the value proposition of cell and gene therapy is really, it's important that it's there, that it makes sense to make investments in these therapies? Maybe Faraz, you could start with that one.
Great.
An easy one.
Yeah, exactly. The cost of therapy and how do we... So I think this is another area where Tenaya's strategy is a little bit differentiated and sets us up in a different way. So for the diseases that we're focusing on, these genetic forms of heart disease, and they are standard of care. Some of the small molecules and in some cases generic therapies are being tried and used, but they're really palliative. They're symptomatic relief. They're not really addressing the underlying cause of the disease. So in some ways there is really no comparator because they're not effective. They're not. So the lock and key gene therapy approaches that we're advancing are in first in class, opportunity to be best in class and life-saving. And so there's going to be hopefully a medical value that will be distinct and differentiated versus anything that's available today. Of course, the gene and cell therapy field, like other organs, for the eye, for the liver, for the brain, for the muscle, everything that has been learned, and for oncology, everything that's been learned over there that you have to demonstrate. First and foremost, it needs to be a dramatic effect. It needs to be a durable effect. It needs to be safe. You need to be able to demonstrate that you're offsetting some medical costs, that you're improving quality of life. All of those things go into the math of a payer and the health economist. So we'll have to do that just like everybody else, but the starting point won't be that our therapies will be compared against, say, a generic antihypertensive or ACE inhibitor or ARB. So I think that's a good starting point to be at. In some cases, the right comparator is actually a heart transplant. Right? Some of the diseases that we're working on, that is the only option that these patients have ultimately when their hearts progress to heart failure, and those are expensive. In fact, a heart transplant is one of the most expensive medical procedures in the U.S. healthcare system today. A heart transplant can cost upward of $1 million. And in the case of a child who is dying of a genetic disorder, they may actually need 2 or 3 transplants as they grow over time. The other comparables might be an LVAD, which are also a couple $100,000. So I don't think the comparator really will depend on the specific disease that you're working on, but we have to do the hard work of demonstrating value, just like every other therapy has to. It's going to be a little bit harder for therapies intended for large indications, because that's where the payer sensitivity on the budget impact and the cost impact will be much higher. But for rare diseases, I'm not suggesting that we want to price it responsibly, but I think there's more opportunity to demonstrate value and more perhaps forgiveness and permission to be compared against something other than the more generic small molecules that are out there. So that's Tenaya. That's somewhat distinct perhaps than what might be true for more prevalent indications.
Jordan, what are your thoughts here? How do you make a therapy sort of more attractive than LVAD or transplant? You're in that space.
Yeah, I mean, I think, to pull from some conversations we've had here, as Jochen was really talking in our pre-session about having really, I can't remember the exact word you're using, but transformative care. These can't be small steps forward. These technologies really have to be transformative in what we're able to develop. And, what Michael was really talking about is we started at 50% mortality over 5 years, and we're still there. So the drugs that are coming online are great. But as Faraz says, they really are treating the symptoms and not the root cause of the disease. So you've got to, how are you going to be bringing that forward? We'd love to be developing therapies that really completely eliminate life with an LVAD. I mean, LVADs are wonderful life-saving technologies, but quality of life on that LVAD is terrible. You're constantly in and out of the hospital. The cost is very expensive to everybody. Not a great situation to be in. I think that the comparator outcomes of a heart transplant are actually kind of interesting, but for us, it's extremely expensive. How can we, send those patients on a different trajectory? And I'm just really optimistic with the different technologies on this panel to be able to effectively, treat those patients.
Yeah, and I think there's another perspective here too, in that there's an opportunity cost with LVAD and transplant. Like you said, these are not benign interventions. You are frequently hospitalized post-LVAD transplant. Transplants, there's just not enough to go around. So, I think that there's that calculus that comes into play as well. Mike, what are your thoughts here in terms of what do you think the payers might be looking for? Something.
Well, clearly you have to change the trajectory of disease. The last 30 years of, albeit improving our treatment of symptoms, is just not acceptable, and it's not moving the needle in terms of quality of life or longevity. And that's why our focus is on remuscularization. We think we need to go to the root cause. Now, you could say the root cause was MI, and we're not actually directly addressing that, but the result of that MI was cell death. It's infarcts in the muscle that now result in dysfunction. You slid down the slippery slope of heart failure, and now you become decompensated. Where do you go from there? You can't recover dead tissue. So you rejuvenate, you replace with authentic human cells. That's our basic strategy, because we think we understand the mechanism of action here. And I think that's really critical. Going into clinical trials with kind of data, but it's a black box as to what your mechanism of action is, it doesn't cut the mustard anymore. And I strongly believe that is one necessary ingredient, along with the clinical outcomes, those that impact quality of life, longevity, and those other factors. Because it's just the case, I think it's now fact, we understand. We can tweak cardiac function parameters with drugs and devices and other products, but we have not been successful in moving the needle in meaningful ways.
So payers are often looking for an impact on outcomes, and that's just the way cardiovascular is, right? We do 30,000 patient studies, and that's just sort of the paradigm for the regulatory approvals. But for cell and gene therapy, do you think we need a different kind of endpoint, a different regulatory framework? Do we need to look more at, you mentioned composite endpoints for us. Are there biomarker endpoints, more patient-centered endpoints that we might begin to think about as perhaps even more important in this space? Joachim, you're looking at me.
Yeah. Okay.
I'll ask you that question.
No, thank you. Happy to take that one. Well, I think Faraz mentioned that earlier. It all comes down to the effect size, right? And we've seen that, especially in gene therapy. If you think, we heard earlier today about hemophilia. Hemophilia, clearly regulators were very quick to change to change the approach, because if you give a replace factor and you're dealing with 1-2% factor level, then you have to show long-term benefit. But if gene therapy is able to show factor levels of 50 to above 100% for years and years to come, everybody understands that the effect will be there. There's no need to demonstrate there. In fact, there is no bleed anymore, so what are you going to measure? And I think in our approach, this is really different, right? It is radically different. We're not going in there to show this incremental benefit. We are really trying to regenerate activity of the heart, and the effect size has to be very significant. And I'm convinced that, yes, biomarkers will help us and guide us in the early development to make important decisions. But I'm not afraid of showing the even pivotal endpoints, because we know if you have that sort of effect size, you will see the outcome. And so I'm very much looking forward actually to those regulatory discussions, because I expect them to be quite supportive.
That's great. That's really exciting. You have lots of confidence in that. It's very terrific.
I already spoke to this earlier, so I won't
go
repeat
ahead.
what I said earlier, but I think there's somebody who's not on the panel here today, but if Verve Therapeutics, which is another cardiovascular gene therapy company, they're taking a gene editing approach to lower cholesterol. And it's, that's a great example of something that could be a one-time genetic medicine approach, where if you can demonstrate, that you can reduce cholesterol, LDL levels, and can do that durably, as they've already shown in non-human primates, that's pretty clear. That's an accepted biomarker. They don't have to go for a long outcome study, because everybody understands that the reduction of LDL, durable reduction in LDL levels, is going to, save lives. And so I think so it really depends on if you're talking about acute MI versus you're talking about congestive heart failure, or talking about genetic cardiomyopathy, or you're talking about, cholesterol lowering conditions. And ultimately, statins, right, are the ultimate cholesterol lowering company. That's going to be a really interesting dialogue, right, where statins are really effective at lowering cholesterol. They're cheap, they're effective. And if you could do it one time durably with a genetic approach, that's going to be a really interesting dialogue, less from a regulatory perspective, because I think they should be able to get an approval, but more, how will payers think about a durable one-time gene editing approach for cardiovascular indication that could have, maybe in a hypercholesterolemia, which is a smaller population, maybe they'll look at it differently. But once you start going to the masses, then it's going to be a very different dialogue. So I think the regulatory strategy and endpoints and discussion about value, it really is going to come down to what's the technology, what's the indication, what's the magnitude of the effect, and there won't be a one-size-fits-all, and that's appropriate.
Yeah. No, I think that those are really great points. So we started this discussion, or one of our earliest points in this discussion was around the patient. Mike shared his family story. That's why we're all in this, is to improve the lives of our patients. I wanted to finish today by asking all of you what you are all doing to incorporate the patient perspective into your clinical development, and particularly what you're most proud of in that area. So Mike, maybe I can start with you.
Well, it's tough for you to put yourself into the shoes of a patient, but what we try to do through the development process, we have something called customer inputs in the device business, and the customers aren't just the surgeons or the operating room staff, they're also the people who live with your product. And sitting down in patient focus groups with LVAD patients and understanding what they like and don't like about their device. Well, I can tell you, there's one thing they like about their device, it keeps them alive. And they'll give you a list as long as your arm of all the things they hate about it. We're in a really interesting position now where we're talking about a one-and-done approach to therapy in many cases. In some cases, maybe it will be repeat therapies, but you're talking about a procedure that once it's refined will be potentially an outpatient procedure. So that's what we're focused on, because carrying around devices and managing your therapy day to day is a burden on patients. If we look at the diabetes analogy, the closed loop systems are changing people's lives now. but they still have to be managed. These systems are insulin pumps combined with continuous glucose monitoring, and they have closed-loop algorithms that allow the system to manage insulin delivery, and it's really a remarkable improvement in the state of the art. But the patients, even children, still need to monitor and manage their device day to day. So as we are developing our therapies, that's top of mind, is what is the patient experience with our product, and not just the magnitude of improvement of their quality of life, but how can we minimize the burden of their disease?
That's great. BJ?
I'm going to take a little different angle and talk about it from a clinical development perspective. We're working in AMI, it's essentially a trauma. And, we have sometimes difficulties enrolling patients, right? Because the standard of care now is quite good. You have an intervention early on and patients feeling good. How do you get them into the study, right? So we spend a lot of time in clinical development understanding kind of that patient flow and that experience just following the AMI to help us think through how we can bring these patients in and convince them to be part of our studies. In some of our studies, we think about quality of life, so we build in those kind of endpoints as well, and so we do get some direct feedback from patients over time to understand this. And of course, we try to understand the patients more holistically through a variety of different means of gathering information and so forth.
Jordan?
Yeah, I think it's a great question. It's really easy to talk, it's a difficult conversation. It's easy to talk about mechanism and get wrapped around in that and what that product is and how you're doing your manufacturing and how, but at the end of the day, there's this interaction with the patient and not just that patient, but the patient's family, that care network around. And so really understanding them, their needs, and how this is going to be, discussed with. We work with patient advocacy groups and really, try to understand that, the disease process, the support networks that are needed for those patients and where you can be interfacing with them and really just capturing as much information on that patient and their needs as possible.
Jochen?
Yeah, so we at Blue Rock, we are experiencing that already. We have an active ongoing trial in Parkinson's disease. And you have to, I know it's very difficult or almost impossible to pretend being a patient, but you have to think about the decision that a patient has to go through. This is not a small molecule where I take a pill and worst case I wait a day and the drug is gone again. Once you take this cell implant, you have it for life. There is no way back. And so we have to realize, and we do that by speaking to the patients, that this is a very important decision. It is similar to receiving an organ transplant and understanding that the patient has to make a very challenging decision and walk through the whole consenting and process. But so far, I think if you do it right, this is well appreciated, and clearly you work with advocacy groups and outreaches, but you're absolutely right. It's a very important piece of that puzzle.
And for us?
Yeah, I mean, I cut my teeth into this industry at Genzyme, which was one of the original rare disease companies. So patient centricity, I've still yet to see that done par excellence in the way that it was done there. And so that runs deep in my DNA. But many of the same things that the other panelists have mentioned, we're years away in some cases from some of the INDs that we're going to file, but we're already engaged with those patient advocacy organizations. We just presented the board of one patient advocacy group. We're bringing them in to speak. We're capturing pictures and videos and stories. Some of it is internally for our own people. Some of it is to raise awareness for these forms of genetic cardiomyopathies and monogenic disorders, which are not well understood. So that is, in some ways, I would say, part and parcel of a playbook that has existed for 2 or 3 decades, and that way we're not special or unique, but we take it seriously. It's really important to us, and to understand that patient experience and what's going to move the needle to them. Because very often the insight is that the treating physician will have a very different perspective of what is going to be meaningful and acceptable to a patient. But when you talk to that patient, or as somebody said earlier in an earlier panel, maybe we should be talking about them as subject matter experts. We should be talking to those subject matter experts. I love that you asked the question, what are we proud of? What I'm really proud of at Tenaya Therapeutics is our first ever patient advocacy hire is a subject matter expert. It's actually a woman who herself has a genetic form of hypertrophic cardiomyopathy, and it runs in her family, and she's lost family members. And what I was looking for is an email she sent to me recently saying, thank you for taking a bet on somebody who really is more of a patient than an experienced professional patient advocate, and building patient advocacy around me. So I'm deeply proud of the fact that we're voting with our feet by bringing an actual person into the company to be the voice of the patient, a true voice of the patient, because she is one herself. And she's been engaged with these patient communities around the world for years, not as a pharmaceutical representative, but as somebody, as a mom, as a daughter, as a sister. And what better thing could we do to bring that voice in our organization and hold our feet to the fire, whether we're talking about trial development, informed consent, cost of goods, and eventual price. We've got somebody who's going to be keeping us honest, and what more could we ask for?
That's great. Well, I definitely love that phrase. I'm going to use it more often now, subject matter expert for our patients. So with that, I think we're at the top of our time here. I want to thank our distinguished panel here. This has been a fantastic discussion. I look forward to incredible progress in all of your programs, and maybe next year we'll be hearing some results, so that would be great. I want to thank the audience for your time and attention, and have a great rest of the meeting. So thank you.
Thank you, Monica. Thank you.